Welcome, everyone. This is another exciting installment of the great research we have going on in the department, and we're going to start with Doug Woodhams introducing Brandon.
Hi, everyone. Thanks for coming today. It's a pleasure to introduce Brandon LaBumbard, who is an undergraduate at SUNY Oswego and went on to do his master's at Southern Illinois University, and also in part in Peru, where he got to do some cool field work in the rainforest. And so he came already with a lot of field experience. And you'll hear about some more of his field experiences today in Vermont, and some pretty exciting results. So I'll let you take it away, Brandon.
All right, thanks. Share my screen. Okay, everyone can see this now? Okay. Hopefully. So today I'm going to talk about the global change ecology of amphibian Chytridiomycosis, specifically dealing with microbes, mucosal defenses, and multiple hosts. And we're going to start our journey today going over the four main regions of my my dissertation, where first we'll start with the nice epidemiological triangle and some broad background information. Okay. So this is your standard epidemiological triangle, where you have a host, pathogen, and environmental interactions that all coincide together and sort of really interplay with each other to drive a disease system forward. Because without a host, you cannot have a pathogen. That virus or microbe or protozoan just exist in space without being pathogenic. It's not until they find a right host that can actually be infected and subsequently become a pathogen. And all this is interplaying with the environment that causes that pathogen to be able to infect that host and cause transmission. But that host also has to become susceptible through these different changes in the environment to actually become infected as well. So you can picture this as a triangle in its sense. But once a host actually becomes exposed to a pathogen, there are a lot of behind the scenes type networks that are occurring. They're activating their immune system to try to fight off that infection. And then they also have their microbiome, either their internal or external microbiomes, that are also helping sort of combat this pathogen to help clear themselves of infection or maintain that infection and be able to tolerate it long term. Okay, but this also is not existing in a vacuum. Here, those microbes, that microbiome, is also part of that pathogen network. So that pathogen, whether it be a bacteria or a fungus, are also part of microbiome of either the environment or on that host. And then because of some type of perturbation, that fungus or bacteria then can become pathogenic and cause disease. Okay, so the microbiome, here we're calling this the collection of microbes that exist in an area, tissue, or organism. These mostly deal with the fungal community and the bacterial communities that exist, but it could also be the archaea and viruses as well. And just looking at the general history, we started going down this avenue a long time ago with just culturing these microbes because sequencing wasn't invented yet. So we were able to basically see some type of change in the environment and under stable conditions grow that microbe and then study it from there. And that sort of got that ball rolling on studying the microbiome. But then in the early 1970s or so, sequencing became, that technology started to be developed. So that allowed us to explore the realm of microbiome a little bit more because you could then sequence the things that were not culturable, especially in the sense that 99% of microbes currently cannot be cultured using available methods. And just a little side note, that's for mostly soil and marine cultures. But if we're talking about like hosts like amphibians and stuff, we did culture roughly 97%, so a little bit better than the soil microbes. But really that targeted sequencing really opened up the avenue for exploring all these different interactions that are occurring within that microbiome. Especially once we got to metagenomics with next generation sequencing, we're really able to see how genes were changing and how one microbe was interacting with another through correlation matrices and stuff like that. So down here, this figure that is displayed here is just a nice bar graph starting around when next generation sequencing really started to take off around 2000. And subsequently the years following up to roughly right around today, with the number of microbiome studies that just from PubMed searches that have currently been explored. And you can see that these are roughly around 4,000, between 4,000 and 4,200 studies just last year alone. The majority of this green bar is made up of human studied microbiomes, since that's really where, for human health, most of the studies are going to go. But specifically, just with amphibians, you can see the little bluish top of the bar here, where starting in 2007, that's when the first microbiome was sequenced for an amphibian system, and that's been increasing over time, largely in part to many members of our lab who have contributed to this knowledge. And currently to date, there are 253 amphibian microbiomes published since 2007, with one of those being one of my first publications just being announced today. So, okay, so looking at this microbiome, we can really study it to see how the changes in the composition can result in the effects on host health. And this could be linked with dysbiosis, or the disruption of the microbiome that sort of leads to more of a chaotic environment where it's not really, there are no similarities between different samples. But then also looking at like resilience and stable states where from a perturbation, you're able to change or go back to a microbiome after that disruption. And then also functional redundancy, where you could have certain microbes that sort of take over that niche that other microbes that disappeared have been fulfilling. So really, for the first part of my dissertation, I'm really looking at how this culturing technique allows us to look at the direct interactions with a pathogen. And that's especially important given that the fear of fungi is really taking hold, where we're seeing a really sharp increase in the number of pathogenic fungi showing up, specifically tailored towards wildlife diseases. So here, this was the cover of Nature magazine for the journal for Nature that came out a couple of years ago, really highlighting Chytridiomycosis in amphibians, and also talking about how in other wildlife systems and also human health, we're seeing a rise in the number of fungal pathogens. Okay, so for the majority of my talk today, I'm going to talk about two fungi in general, but for my first part of this talk is really going to be highlighting several different wildlife pathogens. So here, I'm just going to introduce them. So we have Batrachochytrium dendrobatitis, also known as BD for short. It's way easier that way. But that's affecting amphibians on a global scale, shown up here in the top left corner. So it mostly infects amphibians in the frog realm, So mostly frogs are susceptible to BD. And this has been found all over the world. And then more recently, we have Batrachochytrium salamandrivorans, which is known as the salamander eater. It was first discovered in Belgium a few years ago, and has subsequently been spreading since then across much of Europe. And this one is mostly affecting salamanders, hence why it's called the salamander eater fungus. And it's of particular interest for us here in the United States because the United States is a biodiversity hotspot for salamanders, where we hold, I think there's roughly 700 of the 900 or so species of salamanders found here in the United States. So if this fungus was introduced here, it could be a big problem for our own amphibian biodiversity. And then we also have Fusarium keratoplasticum, which was first identified as a crop pest for many pea farms, I think, pea and wheat. But it's mostly a pathogen for culturable crops. But it can also be infectious to vertebrates. So it was isolated from a die-off of harlequin frogs in Panama a few years ago. So we decided to also look at that. There have also been studies of immunocompromised people and rabbits and stuff that are also susceptible to Fusarium type infections. And then we also have Mucor. This is also a crop pest causing blights of several types of crops. And it also pops up in the aquaculture realm several times a year or so. So Mucor has also been known to infect fish and cause die-offs there. And then down here in the bottom left, we have Ophidiomyces ophidiocola, which is a naturally occurring soil fungus that really since around 2013 or so has been popping up in both the snake communities for hobbyists that have snakes in captivity, but also more recently in the natural environment where several vipers, like the eastern rattlesnake, eastern diamondback, and timber rattlesnake, but also other commonly found snakes like black eye snakes and stuff. So it's becoming more of an issue for wild populations of snakes throughout much of the eastern United States. And then this last fungus is Chytridiomycosis destructans, also known as white nose syndrome in bats. That was first introduced right around the mid 2000s, around 2006 or 8 in New York. And since then has spread up into many parts of Canada and all the way over to Washington State and down to Texas. So it has really, really ballooned since it was first introduced. And it has caused like the declines of millions of bat populations in the cave-dwelling bats, specifically the little brown bat that is highly susceptible. Okay, so these 6 fungi really take us into the first part of my dissertation, where I'm looking at possibilities for inhibiting these pathogens using bacteria. So volatile organic compounds are, just gotta move this picture over, so are produced by bacteria, and these are for sort of a long distance chemical warfare. So the bacteria is able to secrete these compounds that can be aerosolized and travel long distances to sort of inhibit the growth of other microbes around, giving that bacteria more room to grow themselves. And most of the literature on this topic stems from plant and soil microbe interactions. So that's what you see here. There are several studies just highlighting that these soil bacteria are capable of producing volatile organic compounds to inhibit pathogens, specifically fungal pathogens. And then we also see these VOC type compounds also being used for chemical recognition in several animals. So here we have this Hypsoboas tree frog that through these skin secretions, VOCs are being produced, and they act as like a individual recognizer for mate selection and stuff. Okay, so these VOCs have also been recently been studied working with the inhibition of white nose syndrome in bats, where one bacteria that was isolated actually from a banana was used for inhibiting that fungus. And then they also, right around 2015, actually used some of the metabolites that are being produced, these VOCs from that bacteria, to treat and clear infected bats. So this was sort of one of the first successful wildlife stories of releasing bats in the spring that were able to survive infections throughout the winter using this VOC producing bacteria. Okay, so what I did was take 43 bacteria isolates from amphibians. These were known anti-BD bacteria from previous studies. And I really wanted to see if these bacteria were capable of producing these volatile organic compounds. So to do this, I took the bacteria and spread it on one side of a Petri plate. And then it had a division, a plastic division, so there was no direct contact between bacteria and fungi. And then we sealed those plates. And then after a week, we were able to see if there was actual inhibition of that bacteria causing VOC inhibition of the fungus. So here you just see on the one side, there's this yellow bacteria. And on the other side is where that fungal growth could be measured compared to the control. So we were able to take images of those and then basically using ImageJ, calculate the percent surface area to act as a measure of growth. And then using those values, determine if inhibition of the fungus was possible. So you can see here, there's clear growth inhibition from this bacteria. So this is the chytrid growth here. Okay, and then once we collected these results, we're able to then see which bacteria could be potential candidates for future trials. And to do that, these future trials could include inoculating soil to see if you could inhibit the fungal growth. But you also could send those bacterial samples away to perform mass spec to see what compounds are actually being produced, to see if any of those are known antifungal agents in general. So that's some future work that our lab is working on. But here are the results from that study. So what you see here is each panel here is each of those 6 fungi. So we have BD, B-Sal, Fusarium, the mucor, the Chytridiomycosis, and the Chytridiomycosis fungi. And these are all the 43 bacteria isolates that we used. And the colors represent the bacteria that were significantly inhibitory against each of those pathogens. So you can see the majority of those bacteria were inhibitory against the 2 main chytrid fungi, BD and B-Sal, which is not really surprising. These bacteria were inhibited from or cultured from the skin of the amphibians, so it made sense that they would be more inhibitory against amphibian pathogens. But what's really interesting is that the Fusarium and the mucor are sort of resistant to a lot of those those bacteria. So they're pretty tough fungi. I did some other trials with the Fusarium, and there were a lot of antifungal agents that pretty much did not do anything to inhibit the growth of it. So it's a pretty resilient fungi. But when we're looking at the Chytridiomyces and the Chytridiomycosis destructans, we also see a lot of broad scale inhibition from those bacteria, VOCs. Okay, so now we decided that potential candidates would be ones that have broad scale activity against multiple fungi, and also would be able to inhibit at that 80% mark or more. You can see here that most of the ones inhibiting BD and B-cell are right around 100% inhibition. So it's, when we're picking candidates, we have a large pool to pick from there. So in general, just from this study, we see that these 4 bacteria were actually able to inhibit 4 of the 6 fungi by at least 80%. So those would, except for this one was actually inhibiting the Fusarium fungi and also 3 other ones. So I decided to include it in potential future probiotic trials for just because it was one of the only that was inhibitory against Fusarium. And so what we see here are these four bacteria that could be used later on for future probiotic or bioaugmentation trials. And what's really interesting is that if we use these bacteria, we could potentially have broad scale activity for inhibiting multiple fungal pathogens in the wild. So a nice way to tailor our conservation efforts and mitigation efforts for future trials. Okay, the next part, this is the main chapter of my dissertation, looking at the leopard frog microbiomes across the United States, and really trying to link disease and climate change to those changes. So here we're looking at overall seasonal patterns in the microbiome that shift due to the changes in temperature and also the frog activity. And to do this, we have 5 locations across the United States. Up here, the field site that I actually monitored up here in Vermont. And also we have colleagues that worked in Pennsylvania, Tennessee, Louisiana, and New Mexico. This is part of a startup grant looking at Department of Defense lands for amphibian population monitoring. And so what we did here was 2 times every season, so spring, summer, fall, and winter, we would go out and survey leopard frog populations, collect them, and basically look at their disease status for BD and B-cell, but also monitor their microbiomes through time. Also, just for these 2 northern populations, we don't have any winter samples because they where the frogs would be under the ice hibernating. So we only sampled them for spring, summer, and fall. Okay, and this goes back to our epidemiological triangle here, where we're really trying to see this interplay between the environment, the amphibian host, and the fungus. So here you can see this is BD growing in culture. So it's starting out as a sporangia that then releases these zoospores. The zoospores are the reproductive stage that are actually what is infecting the skin of the frog. And then within that host, we have different immune functions like antimicrobial peptides and antibodies, and also those beneficial microbes or the microbiome on their skin of the different type of bacteria and fungi that are coexisting on the skin of the frog. Okay, so why study the amphibian microbiota? This is mostly for the mucosal defenses that we're seeing on the skin. So there are currently 8,236 as of today species of amphibian documented or known to us. And roughly 700 of these have been negatively affected by chytrid over the last several years. So yeah, it's important to try to look at those to see sort of what we can do to mitigate that disease. And how are we doing on time? I actually can't see time. Okay, there we go. So by looking at this outer mucus layer of the frog where the bacteria coincides, that's also the first line of defense for those infectious zoospores when they're trying to burrow into the skin and become infectious. But then within that skin, we have B and T cells that are actively trying to fight off infections. And then the granular glands where peptides and more mucus is being produced to be secreted onto that outer mucal layer. Okay, oops. So once we're in the field, we would come across the frog and capture it and swab it to collect the genomic material that we then were able to extract and perform PCR amplification to amplify the cytosine rRNA gene for bacteria, or the ITS transcribed spacer region for fungi. And then we are able to sequence that here at UMass using an Illumina MySeq, and then perform data analysis using a pipeline called CHIME for analyzing microbiome samples, and then also export that data to perform statistical and data visualization in R. And then from this genomic DNA here, we are also able to test to see if that amphibian was actually infected or not with the BD pathogen. All right, and some preliminary findings. So as of right now, I've only completed the 2017 and 2018 sampling. I still have the 2019 samples to process. But so far, what we're finding is that the alpha diversity, or the within sample taxonomic diversity, is actually very different across those 5 different sites that we're studying. Here you can see this is colored by the species of leopard frog that we're finding at those locations. And there's really no similarities between those species. So in Vermont and Pennsylvania, we have northern leopard frogs, and you would think that as a species, they should have similar microbiomes, but they're actually quite different. Same with southern leopard frogs between Tennessee and Louisiana. But once we have that microbiome data, we can then filter that data to sort of show the general trends in the microbial community. And that's what I'm showing here with this bipartite network. So here, these are showing some main nodes, so a main node for each of the 5 populations. And what we see here is that there are several bacteria that are being shared across communities. So the Cyanobacter, Commonwealth they're all being shared across those locations, but some populations have a higher relative abundance compared to others. So the thicker the line, the higher the relative abundance in that sample. And so what we see here, just to quickly go into a little bit, one of the important parts is this Githanobacterium. So this is a known group of anti-BD bacteria. What we're seeing here is that it's really only highly abundant, above 1% relative abundance in Vermont and Pennsylvania. This is also known as a cold-loving bacteria. So it could also be that in these more northern ranges, we're seeing like that bacteria being more abundant because it's just more prevalent in the northern populations. Okay. And then here, this is just looking at the percent anti-BD bacteria. So we can use our microbiome and sort of cross-reference that to a known database of antifungal anti-BD bacteria to see what proportion of our microbiome is actually anti-BD. And what we see here, the general trends are that there are seasonal differences that vary across the 2 sample years between from winter to the subsequent following fall. So many seasonal differences in most of the populations. But then if we're looking at frogs that are actually infected or uninfected with the BD fungus, we also see differences there, that infected frogs seem to have a more inhibitory microbiome. Okay, so this is just the bacterial beta community, beta diversity. So looking at the overall trend in across samples. So here each dot is representing one individual frog sampled. So the distance between the plots points is showing one community that's more similar to another, or if they're further away, they're more unique from each other. So here, looking at the Spray-Curtis index, or looking at the OTUs that are present, or the bacterial species that are present in their relative abundance, we can see that there's very big differences in those two sample seasons. So interesting. Okay, we also sampled the mucosome of these frogs. So basically we took that frog and put it in a water bath and then froze that sample and processed it, lyophilized it to concentrate that mucus further, and then performed a cell viability assay to see like how viable that mucus is at inhibiting the fungus, and then also process that data. And what we generally see here is that as the frogs are more infected, so if they have higher infection loads, we see that mucus is becoming more inhibitory. So that's interesting that we see that trend there. So it's the frog is obviously responding to that infection. But this is species specific, where these northern leopard frogs seem to be really driving that trend in that data. Okay, and then the last part for here is just really looking at, here we have a comparison of the skin peptide community that was also sampled from the same frogs, comparing that to the microbiome. So here the gray lines represent the bacterial community, and then the black line is that peptide. So we're seeing really strong seasonal changes in both the peptide community and also the antimicrobial peptide community with the microbial community that really seems to be driven seasonally. So if there's, we could talk about this for questions if there are any. So, and then the very last part is just really what future work I plan to do. So I still have to finish the 2019 sampling from my field samples. And then from that VOC work, the next step is to take those sequences of those 43 bacteria and sort of compare them across multiple wildlife hosts to see how prevalent those 43 bacteria really are in different communities, to see if this broad scale type inhibition could actually take place for future trials. And then the very last part that I'll touch on very, very generally is that we also have some work where we're sampling for the two BD and B cell fungi in museum specimens, and also looking for non-amphibian hosts for those fungi. So so far we've swabbed turtles and salamanders across much of Massachusetts, and some museum specimens of salamanders from Laos, and then also crayfish from many parts of the United States, just to look for overall similarities in BD or B cell detection. And then for some lab experiments here, I took crayfish and exposed them to BD and B cell just to see if they were actually infected or infectable, and found that there was increased mortality when they were exposed. So we're going to be doing some future trials to look for possible infection, and also take those exposed crayfish and see if we can induce transmission to an amphibian host to look at pathogen transmission from a non-amphibian host to an amphibian host. Okay, and basically that is what I've been doing for the last several years and have lots of people to thank. And with that, if anyone has any questions, I can take those. Or if we're short on time, we could wait until after the session.
Yeah, I think we have time for one quick question in the Q&A, or we can also ask at the end.
Yeah, thanks so much, Brandon. Really great job, and this is really only a part of the work that you've been doing. So it's amazing to see how much you've already accomplished here. I'm just waiting to see if there's any questions from the crowd, and I'll read them out.
Yeah, so pause this guy.
Okay, looks like there's no questions right now, but we can
Okay.
maybe we can come back to you at the end.
Yeah, perfect. All right.
All right. Great. Then, hi all, Jared Burns. It is my distinct pleasure to introduce our next speaker, Brianna Shaughnessy. Brianna is a PhD student who's been in my lab since 2017. She comes to us by way of doing a bachelor's at Wheaton College. And then after that, she was a student in the 3Cs program and did a master's in professional science at Northeastern University, looking at color polymorphisms in isopods. Some really cool work. I was really lucky that at the end of that, she came and was a diving technician in my lab for the summer, did some awesome work. And I'll admit, the time I was like, nah, I'm not gonna take any more students. But she just so bowled me over with how amazing she was that I've been delighted to have her in the lab for the past few years. And just to show you how amazing Brianna is, currently she is a NAUS fellow working in the National Oceanographic and Atmospheric Administration Office of Education for Aquaculture. And in fact has been offered a contractor position with NOAA next year. So she'll be skating the line and both completing her PhD as well as working for NOAA. So like just someone who can knock it out of the park and does it all. And I'm continually blown away by the work she does and how she thinks about the world. So with that, Brianna, take it away, and I'm delighted to hear about what you're going to tell us today.
Thank you, Jarrett. I'm going to just share my screen here. Okay, awesome. Thank you. Thank you all for coming today. I'm excited to share what I've been working on for the past couple of years. This might be a little bit of a different seminar than biology is accustomed to, but I'm going to work with the 30 minutes that I have to really explain the development of this transdisciplinary dissertation and the chapters that really try to address the questions that we've developed along the way. So in order for you to understand why I'm taking the approach that I am, you kind of have to understand where I come from. For one, I grew up on Cape Cod with water women and water men in my family. And when I joined Jarrett Burns' lab in the summer of 2016 as the diving technician, I was kind of in that existential crisis that happens after a master's degree. But I got to work on the kelp ecosystem ecology network, also known as KEEN, which is a large-scale collaborative effort across the world. As you can see on this map, it's an incredibly powerful collaborative research effort. The coordinated sampling design for annual data collection allows subtitle ecologists to track both regional and large-scale temporal shifts in the community assemblage of kelp ecosystems all over the globe. Each of these highlighted areas represent a varying level of sampling sites all across those countries. So working with KEEN, I was able to chat with New England ecologists about the shifts that they were starting to see in some of the kelp ecosystems around here. And then in reporting out what I learned to my family, I also started to hear about this great new aquaculture crop in New England, which was seaweed, mainly kelp. So I started to wonder really how we might connect what we're learning about kelp in these natural forests with what the kelp farming industry might want to know. And luckily the UMass-Boston Integrated Graduate Research and Education Traineeship was going on when I joined. And also luckily Jarrett was an IGERT fellow when he was a PhD student. So this fellowship is really geared towards scientists like myself who are looking to break the boundaries of a traditional PhD dissertation and really develop research that works within communities to meet their needs in working to solve complex environmental problems. So why does seaweed farming fit within this transdisciplinary approach? Well, for one, seaweed farming requires this deep understanding of not just environmental biology and ocean and coastal processes, but also coastal and marine policy, community level needs, and even business management at times. Globally, seaweed farming is not a nascent field. Some forms have been practiced for centuries. And as you can see with this graphic here, over the past couple of decades, we've seen this shift from harvesting seaweed in the wild at small scales to cultivating it at large scales for commercial production. In the United States, though, seaweed remains a nascent industry, but it is the fastest growing domestic aquaculture sector at about 8% and still rising. And we seem to have kind of this true buy-in for domestic seaweed as a sustainable crop, but we really need to kind of capitalize on this early phase expansion opportunity where we can really think about where we're directing our research and our resources in upscaling this industry and trying to do it in a truly sustainable way. And if we can do this properly, we might even be able to meet this triple bottom line of economic, social, and environmental sustainability. So there are a handful of seaweeds that are cultivated in the New England region. Here's a few of them just to show you. Ecologically speaking, the two species that you see in the middle here are keystone species for coastal subtidal habitats around New England. They provide food and shelter for fish and invertebrates. But the species that we're talking about today is Saccharina latissima, or sugar kelp. This kelp loves rocky bottoms and a good amount of wave action, and its ability to grow rapidly under rough conditions really makes it a good crop to grow in the winter months when business is slow for other commercial marine sectors. There are a couple of different cultivation techniques for sugar kelp, but generally speaking, this is the most common. It's grown on long lines that are suspended horizontally just below the ocean surface. And in this schematic, you can kind of see the long lines are suspended horizontally and attached to anchoring buoys on either end. And then those buoys are anchored in the sediment on moorings and chains. And what that looks like in real life, what you're seeing here, what you saw on the long lines actually were juvenile sporophytes that have been transferred from aquaculture hatchery facilities that create seed strings. And you can see an example of seed strings in the top photo here. Seed string is really just tiny little sporophytes that are attached to a small string, and that string is wound around the larger ropes that are hung horizontally in the ocean. The kelp blades grow throughout the season using natural light and available nutrients in the water column to grow, and then they're pulled for harvest, usually late spring or early summer. And then those blades are sometimes dried before processing. Other markets use the wet weight or the wet blades when it goes to market. So sugar kelp itself is rich in vitamins, B vitamins, iodine, and other vitamins and minerals that make it really great for human consumption. The market spans for snacks for direct human consumption to dietary supplements, sometimes fertilizers, animal feed, and the byproducts are also used for thickening and gelling qualities in both beer and biomedical supplies. But as you might imagine, finding spots to put those long lines can be really tricky. In Maine, they have a fairly streamlined permitting system, but Massachusetts itself does not. So farmers that are looking to enter the industry are often left to do some digging on their own. they need to understand the potential environmental impacts of their gear and the physical and chemical oceanography of their sites. And at this time, what usually happens if a Massachusetts farmer is looking to get a new farm going, they'll look at what has been or can be permitted in that area for aquaculture in order to choose the right spot for them. But these are not characteristics that are specific to seaweed. They're often specific to things like oysters and other shellfish. So my first chapter here is really an effort to understand what information we can prioritize to help farmers really scale their farms with seaweed-specific evidence-based practices. And we're looking at this kind of through the lens of a review of ecological and environmental data. So what do we already know that might lend a hand to understanding where to farm seaweed? How can we leverage research to fill in gaps in what we know? And how can we use the knowledge that we're gaining to understand what we can avoid as we upscale? And a lot of this was actually born out of conversations that I had with the farmers themselves as I started to kind of look into this research topic. So our first step here in this effort was a literature review of existing data on the abiotic factors that are affecting sugar kelp growth in natural communities. So marine ecologists generally cite about 16 degrees Celsius as the survival threshold for Saccharina latissima. And researchers like Val Gerard have been investigating these thresholds for many decades through observational and manipulative experiments. And so we wanted to look into that data and see what we could pull out of there. So we searched the Web of Science database for available peer-reviewed literature, and we found about 22 papers that met our criteria of sampling either cultivated or natural sugar kelp, reporting growth or biomass yield, and including metrics of temperature and nitrogen concentration. So we're able to create an effect size by pulling all of the data out of these papers, and we created a binomial regression of that effect size of temperature on sugar kelp growth rate per day. And here's what we found. That 16 degrees Celsius threshold does seem to ring true, but growth rate itself kind of peaks at effective temperature right around 10 degrees Celsius. So a couple of things to keep in mind here are that survivorship thresholds might be relevant in ecological studies, but growth rates are really of most interest when you're talking about industry interest. And secondly, temperature is not the only factor, as we know, that affects growth in marine ecosystems. We have nutrient availability, which we did try to pull out of these papers, salinity, pH, and many other abiotic factors to consider here, but we'll get a little bit into that in the second chapter. But in terms of obtaining that data from the research that existed, we found some real gaps in consistent research about these things, and we really weren't able to pull that much that was usable here. So that started getting us thinking about how we can begin to fill those gaps in what we found. And one way to do so, KEEN itself is working to understand the impact of global change on the world's kelp forest that includes sugar kelp in this area. If we can start to connect the industry with this goal, we can start to kind of expand the usability of the data that we're collecting and maybe even alleviate some industry burden as we move forward. So for one, if we can understand what's happening in the natural sugar kelp populations, we can begin to kind of predict how shifting oceanic conditions like temperature and nitrogen might affect the industry itself moving forward. And there are other important environmental interactions beyond that we need to really consider when we're upscaling this industry. We're manipulating the environment here, and we're turning an ecosystem quite literally on its head. So we're growing something that's usually in benthic communities on long lines suspended from the ocean surface. So as the industry expands, it's going to become more and more efficient, and we need to keep in mind what we can learn from the past that we might be able to avoid. And agricultural expansion is kind of the quickest link to start to consider. These aquaculture operations really need to kind of prioritize a holistic understanding of the natural processes that are interacting with their crops. And they really, just like soil, need to deeply understand what the waters around their farms can produce sustainably. And we have a chance here to really kind of avoid these catastrophic failures like arsenic in the rice industry, if we can acknowledge the lessons that we previously learned from them. And this is something that we're trying to keep in mind with our future plans for this kind of investigation into what exists out there. We learned that there are water quality resources out there. We just need to gather them. And in gathering them, we're able to pinpoint kind of gaps in necessary knowledge. And in the future, we're looking into kind of these sustainability claims and the ability of farms to work sustainably within different coastal sites. And we're also interested in the abiotic factors that are influencing farms in comparison to how they influence natural kelp forests. And we're looking to do this by creating a research agenda that's targeted towards the New England sugar kelp industry that the industry itself can point to when it's planning different tests on their farms. And then we're also creating opportunities for industry stakeholders to connect and share their perspectives with scientists, both at conferences and with informal networking calls and things like that. But I'll talk about that in a minute. So for now, in the next chapter, we're really attempting to tackle one of the gaps that we found in knowledge and kind of figure out what data we can collect and share moving forward. And I really worked with the industry here to determine questions that we could conceivably work together on by leveraging resources. And this second chapter really attempts to narrow in on the things that we found were kind of a nexus at which we were both trying to find information. Reframed slightly differently, how can we address these gaps to help the Massachusetts sugar kelp industry move forward? These folks don't have time to answer these ecological questions that they see on this slide here, but they certainly are invested in understanding them and understanding where the heck they can grow this stuff. You know, in the process of gathering all of this information, we start to understand where in the Massachusetts coastline we might be able to have some successful kelp farms. One low-hanging fruit in trying to figure this out was our Keene sampling sites in Salem Sound. Our annual sampling seemed really like a good place to start some preliminary research, and I'll get to the farmed sites in a minute, but Salem Sound has some great kelp beds. It also has some great or not so great coastal activities that are relevant to the seaweed farming industry. As biologists, we know that carbon, nitrogen, and phosphorus are our building blocks of life. We can hypothesize that kelp with more access to these compounds will grow larger and potentially quicker. But as we know also, areas with high concentrations of these compounds sometimes end up being areas of high coastal pollution. So in Massachusetts, a long history of coastal industrial activities has caused widespread contamination of marine sediments. These toxic, like legacy compounds, as we call them, persist in the environment long past decommissioning of industries like this coal plant that you see here. And what you're looking at here are 3 kelp sampling sites and their proximity to a sewage outfall called Haste. And also note the decommissioned coal plant that's nearby as well. In this way, kelp's ability to assimilate compounds from its surrounding environment is a bit of a double-edged sword. So due to food hazards implications, I'm gonna kind of focus in on arsenic and lead here. Arsenic exposure can lead to cancers of the skin, and it can also lead to cancers of the lungs as well as heart disease. Lead exposure can cause kidney and brain damage, as well as developmental disorders, but spoiler alert is that brown seaweeds like sugar kelp are particularly efficient at assimilating these compounds. The mechanisms behind them are largely not understood at this time, so the FDA and USDA really struggle to create enforceable standards for seaweed products that are going out eventually for human consumption. So an REU student, Carla, and I started a couple of summers ago to investigate the potential of heavy metal contamination in these sugar kelp blades. And we wanted to start kind of at a small scale to see if this rabbit hole was really worth going down. Taking a look at our resources from the outset, we focused in on the kelp tissue itself. So we weren't able to get water quality testing done at that time, but keep in mind for future research, that's what we're looking to do. So we collected a bunch of kelp from our Salem sites. We dried them at 60 degrees Celsius for 48 hours, ground them into a fine powder, weighed them, and then the UMass-Boston's environmental analytical facility paired with us to analyze samples for carbon and nitrogen with the elemental analyzer, but we also ran our samples through ICP-MS. So you can see in the diagram at the bottom here, we digested our powdered samples overnight in a trace metal grade nitric acid solution, and then we concentrated the samples over heat, which you see Carla doing here, and then we sent samples through to obtain heavy metal concentrations. Circling back for a second to our question, and forgetting about contamination for a second, but our question about how kelp grows in these areas of coastal pollution. Our analysis of stoichiometry found that there was a high level of variability in blade size across these sites, particularly at our Eagle Island location, which was closest to the sewage outfall. Some post hoc tests did find an effect of site itself on blade size, but if we do a quick linear regression on all of the samples, we see that we need to really tighten more into this trend. If you tilt your head here, you might be able to see some different trends going on, but we really need a lot more information to make any claims about this. But what's important to note here is the high level of variability across sites, even within the same body of water. And again, these preliminary tests really helped kind of guide our future sampling scheme. And we can tell here that something else is going on that is worth investigating. But if the quality of the tissue is not safe for human consumption, then the size of the blade really doesn't matter. And just a reminder, we hypothesized that tissue samples that were at sites closest to the sewage outfall would exhibit higher levels of heavy metal concentrations. So this time we started to bring in some samples from actual kelp farms. And again, we were trying to investigate whether this research question was worth pursuing for the industry. Our ICP-MS analysis found no significant effect of sampling site on total arsenic concentration. And there was a lot of variability again here. The white ticks on this graph represent each of the different samples. And upon first look, we might get a little freaked out. The FDA has a standard food contamination limit set for things like lead and arsenic. But as I mentioned before, nothing specific to seaweeds here. So the red line that you're looking at here indicates the maximum contaminant level for arsenic, but this line actually indicates inorganic arsenic, not total arsenic in a sample. And what that means, arsenic and lead are absorbed directly from the surrounding seawater and metabolized through oxidation by kelp and other organisms, along with other compounds like phosphates and sulfates. And there is evidence that there might be a link between phosphate concentration and inorganic arsenic concentration. But kelp can either metabolize total arsenic into non-toxic arsenose sugars, which are organic compounds, or inorganic arsenite. And that is the toxic form of arsenic that, arsenic exposure in mammals uses metrics for. So if we're talking about effects of arsenic on mammals, we're talking about inorganic arsenic. So in order to characterize these tissue samples, we needed to go really one step further and speciate the total arsenic into organic and inorganic concentrations. And this is not cheap. It's about $300 a sample, and there are only a handful of facilities that are able to do this. So we paired with Brian Jackson's lab at Dartmouth College, and he's been researching food hazards for the USDA and FDA for many years, and he's been a great collaborator in trying to figure out this little puzzle. So when we speciated the arsenic, we found that The samples that had the highest level of total arsenic concentration also had the highest toxic arsenic concentrations. So these are important things to keep in mind when determining how to set up environmental analysis for farms that might be looking to be permitted. For example, if there's a high level of total arsenic, then coastal managers might want to spend some time speciating some samples and put some resources into that. It can also be valuable information for this blue economy sector as they try to kind of put a price tag on bioremediation potential for both natural and farmed seaweed communities. In addition to arsenic, lead is the other contaminant of concern here, and lead does not require speciation. Here we did find an effect of site on lead concentration. Of the samples that we tested at each site, one farm and one of our Salem sites exhibited some high levels of samples that had lead content that was over the maximum contaminant limit for the FDA. Again, this is incredibly important information to take into account when we're prioritizing site level testing and emerging farms. And it also helps us understand the environmental services that kelp forests might be lending to us moving forward. So these preliminary tests have really helped guide our plan moving forward. Multiple factors interact to affect the size and quality of sugar kelp blades, and those factors, like best growth conditions for kelp, might be relevant to ecologists, but they're not exactly indicative of a site where we would be able to grow farmed seaweed that would be safe for human consumption. So moving forward, we're looking to have red dots all over this map that represent sites that we've done sample analysis on. We are collaborating with keen researchers, kelp farmers, Brian Jackson's lab, to collect and analyze as many samples as funding will allow. In addition to that, we are adding water sampling for analysis of stoichiometry and heavy metals, just to connect what's going on in the abiotic environment with what's going on with the tissues. We're also collaborating with farmers in Maine that have already done some of these tests and really gathering as much data as we can through both existing and future efforts so that we can help to leverage what we have to model the processes that are affecting the quality of the sugar kelp that we're growing. And then also, as I mentioned before, there's this kind of added bonus of the blue economy and bioremediation information that agencies like NOAA are starting to really get interested in. So the farmers and the researchers in the blue economy sector are, they're not the only ones that are invested in this information. The consumers themselves care as well. This information needs to be disseminated incredibly carefully. So you'll notice that I haven't named a single farm or a single farmer in this presentation, and that's on purpose. My final chapters are really not looking to blow the horn on farms that aren't able to devote resources for these complex and often expensive tests. We're looking to really determine how this data can properly be disseminated and how we can make sure that the audiences that need the data are getting it, and that the other audiences that might want to get their hands on this data really understand the implications of the information that's out there. So I'm going to take a small step back for a second. When we're talking about seaweed farming and aquaculture, it's often paired with other aquacultured products, and sometimes even in co-cultivation techniques where you're growing seaweed with oysters and fish as well. And this word aquaculture means a lot of different things to a lot of different audiences. And also, there's a lot of different ways that you can grow different species that are aquacultured. So all of these kind of lend to a little bit of confusion when we talk about seaweed aquaculture, aquaculture in general. And the perceptions, needs, and practices that vary widely across regions can be a little bit of a hurdle when you're talking to public audiences. And these communities can quite literally determine the regulatory and policy decisions in their area. They can have a huge impact on the acceptance of things like seaweed permitting and expansion. And this makes really the dissemination of information like this a fun challenge to address. And also it requires really ensuring that proper messaging can provide data that's important, but also understood broadly with the general public. Luckily, there are entire networks that have been working on this to understand how to effectively engage the public with conversations about complex topics like these. I just have one example here today, which is the NOKI network. They've been working since 2009 to raise the discourse around topics like climate change and ocean acidification. And they've really perfected this by taking a community level lens and working together to constantly reassess their efforts and figure out best practices to have collaborative conversations and trainings. And I somewhat serendipitously found myself at the center of such efforts this year. I'm currently a Knaus Fellow at NOAA, and I'll soon be, as Jarrett said, transitioning into a contractor position. The Sea Grant Knaus Fellowship matches qualified graduate students with host offices within the executive and legislative branches of the government. And those fellows gain ocean, coastal, and Great Lakes resource information and an understanding of the national policy decisions that are really affecting those resources. This year, I'm sponsored by MIT Sea Grant. I'm their 2020 fellow, and it ended up being a great year for me to apply because the National Sea Grant Office, Office of Aquaculture, and Office of Education came together to host a joint Knaus Fellow to work on aquaculture education coordination. And this is the team that I work with. If you work within aquaculture in New England, you might recognize some of these faces, but these are really the people that have been the driving force behind developing the research that we're doing now. So together with that team at the beginning of the fellowship, we set out to kind of create a plan that would allow us to better coordinate NOAA's aquaculture education efforts. And in this case, education does not just mean K through 12 formal education. We're talking about any effort to get aquaculture messaging out to the public, including efforts surrounding, emerging industries like seaweed. And this past spring, we did a NOAA-wide inventory survey, and we collected 74 responses, allowing us to analyze existing efforts and perceived gaps in NOAA's aquaculture education efforts. Unfortunately, I don't have the time to go through the survey results today because I kind of narrowed into the hard science aspects of my research, but there is a recorded webinar out there. If anyone wants it, I can send it to you. But the survey itself helped us define this problem that you see here that we are working on tackling together and working to develop an understanding of how the efforts that are already existent within NOAA can help kind of push this sector forward. So we have our multidimensional problem that you see here, and that culminates in the need to coordinate better, not just within NOAA, but also across our aquarium and industry partners. So our solution here is to convene those partners to create a community of practice around aquaculture education efforts. So a community of practice is a group of people that share a common interest or passion for something, and they learn how to do it better as they interact regularly, just like the NOKI network did. And what we hope to do is support sustainable aquaculture expansion moving forward by creating able and confident educators that can inform consumers so that those consumers can start to really understand the pros and cons of aquaculture in their region. So I mentioned, up to this point, I really made the case for industry, but I mentioned aquariums here. I'm just going to take a second to explain. NOAA's Office of Education has been managing this Coastal Ecosystem Learning Center network since 2015. And this is a network of 25 of the largest aquariums and science learning centers in the United States. We work with that network to connect and share resources and trainings and best practices. And in turn, the aquarium uses NOAA as a source of credible and up-to-date scientific information that they can disseminate to their visitors every year. Now, COVID has really taken a really hard hit on the aquarium and aquaculture industries, but we've been working with our aquarium member institutions to kind of come up with a plan to support them moving forward. And one of the things that they're looking for is a toolkit of resources to help discuss aquaculture topics with their audiences. And for my dissertation, if I'm going to help my seaweed farmers, then I feel like in addition to my lab experience, I also need to gain experience in science communication, like I am at NOAA, to kind of create these toolkits of resources, not just for our aquariums, but also for our industry partners. So we're working on doing this through a series of workshops. We convened in September for the first time, and these represent folks from across NOAA. So we convened internally to come up with a plan for working with our external partners moving forward. And some of the common threads here, they shouldn't really come as a surprise to anyone, but we found that these misconceptions and misperceptions about aquaculture really stem from the diversity of meanings of the word and a lack of credible, organized pipeline to gather information about the topic. So for example, if someone Googles aquaculture, there's a million different routes that Google situation could go in. Career awareness is another key gap. Aquaculture really represents a great STEM engagement opportunity for youth, and there's really a lack of understanding for the career opportunities that youth might have if they're interested in the aquaculture field. And again, one size fits all does not work. We need community level approaches in order to define target audiences and figure out what communication techniques are most effective for that audience. And if our community of practice can connect often across disciplines, then we can kind of enhance this aquaculture literacy or understanding of aquaculture in the public moving forward. So next week, we're convening again with our aquarium and industry partners. We have 21 states, 2 tribes, 8 aquariums, 3 shellfish growers associations, some restaurant owners, 23 Sea Grant offices, and 9 marine fisheries offices represented in this effort. And we're going to be talking about the audiences that can receive benefits from aquaculture education, how we can push this community of practice forward using different tools and activities. And then we'll be talking about a mini grants opportunity that we're offering to our external partners this winter. So just a summary of our objectives here. We started off trying to review just what existed for aquaculture education across NOAA. We're using that review to design and implement this community of practice that I talked about. And we're hoping to actively evaluate our community's success moving forward and then adapt accordingly with what we learned. In terms of evaluation, we have quite a few metrics to work off of here. I won't dig too far into it because we're running out of time, but we have, like how many people have engaged in the effort, how many people respond to our surveys, how many come to our workshops, how many actually go forward and create some sort of aquaculture messaging moving forward. And thankfully, I have the opportunity to follow this through longer term and really kind of dig into how things evolve over the next year. And as industries expand and administrations shift. And so this has ended up being a really great opportunity, not just to share the seaweed research that I'm doing across multiple audiences, but also to be a part of really building a national effort from the ground up. So I will stop there to take any questions. Hopefully I didn't go over too much. And again, just like Brandon, I have many people to thank. And a sidebar, I moved into Brandon's room when I moved to Boston to start in Jarrett's lab. So it was a great apartment for UMass-Boston researchers.
That's great. Thanks, Brianna. I think we have time for at least one question in the Q&A. And then maybe for both of our speakers, we can move questions to the gathering afterwards, if that sounds good. But take a moment. Feel free to ask something in Q&A.
And Jarrett, I cannot see the Q&A, just so you know.
Oh, you can't. Okay,
No.
I will read you first. So far, nothing is in.
Okay.
Any last questions?
At the bottom where you see like share screen,
Oh, I see now. Okay.
we have one open question.
Yep. From Rick. Rick asked, what is the use of sugar kelp and how is it consumed?
Yeah, so there's a lot of different ways that you can consume or use sugar kelp. So you can either eat it raw if you're feeling crazy. You can use the byproducts, which have congealing qualities to it, to create ice cream, toothpaste, beer, agar plates for biomedical tests. You can also dry it and create kelp jerky. You can feed it to fish. You can feed it to livestock. You can also use it as a supplement in your foods. There's a million different ways that folks are using it. It's just a matter of figuring out if it's safe in large quantities.
Yeah, if you've had kombu before, you've had kombu. If it's from Japan, it's not the same species as here, but it's a congener. And in fact, there are companies that sell Gulf of Maine kombu.
Yes.
All right. I see that Doug has put the happy hour link into the chat. So we've got that. Oh, one last question. Luis, are you asking how much support does the program receive from the government?
What I'm doing right now, or the community of practice? There's kind of two. And so what I'm doing right now is not supported as far as heavy metals analysis. There's no current government support for that. The government's actually a little bit scared to do that at the moment. But as far as the community of practice, I represent the person that is funded with a salary to work on that. So I guess that the level of support is the salary for one human, and that is me.
Fantastic. All right, well, thanks, everybody. We'll see you on the other Zoom line in just a minute. Zoom line, is that what we call them? Yeah.
All what you want. Yes, please come over. Yes.
Here we go. Okay. I'll see
All
you over there.